oxygen, CO2, or you can have ions, as we learned with diffusion, smoke particles, whatever it is, you put it in a compartment that is connected, that has a pathway with another compartment, things are going to move randomly, right? They're going to move because of thermal energy. The higher the temperature, the more movement there's going to be, right? The colder the temperature, the least kinetic energy, the least movement, right? So these molecules are going to hit the walls of those compartments, hit each other, and randomly are going to end up in the second compartment, okay? And this is all random. It just happens because they happen to collide with something and be able to exit through that door. So that's diffusion. It depends on temperature. The higher the temperature, the larger the motion, and it's random. However, we talk about net diffusion, and that's what we've been discussing since the beginning of this course. Net diffusion is directional movement, and it happens due to a higher concentration in one compartment relative to another compartment. So things are going to have move with direction from high to low concentration. We have a diffusion gradient, okay? That could be, that driving force could be a concentration gradient, an electrical gradient, could be an osmotic gradient, a thermal gradient. Anything where we have a difference between those variables in two compartments, there's a pathway. That driving force, that difference, is going to cause net movement of whatever molecule we're talking about, okay? And net diffusion keeps occurring until the concentration gradients are equal, and then net diffusion stops. Diffusion will still continue. That random movement due to thermal motion continues, but directional movement, net diffusion stops, okay? All right, let's talk about what affects net diffusion. This will be the area of contact. So remember, for something to move, we need a pathway and a driving force. Let's say we have that pathway. These two compartments are connected, and we have a driving force. We have a bunch of molecules here, so the concentration is high. 0 molecules here, so concentration is low. So we have a concentration gradient. So things are going to, these little molecules that you see right here are going to move from this compartment to this compartment. That's net diffusion. We have the pathway and the driving force. But that directional movement, that net diffusion, will be larger. The rate of movement will be larger if we have a bigger pathway. So imagine I have these tiny little super balls, right? You guys know the ones that you bounce everywhere, and I have that door. Okay? If I start bouncing 100 balls here and maybe just 2 in the other room, the net movement is going to be in that direction because it's more likely that they'll collide here, all of them, with each other and the walls, right? And end up in that door than the 2 that are over there end up here. It's just more likely. But if now I make that door twice the size, if I open a garage door instead of that, then that net movement of these bouncy balls is going to happen faster. Okay, so diffusion rate increases with a larger pathway. The other thing is the concentration gradient, so the driving force. If the larger the driving force, the larger the diffusion rate. So if you have a lot of those bouncy balls here, 100 of them, and then just 2 out there, then that's going to be a large concentration gradient. We're going to have diffusion occur faster. That directional movement, that net diffusion happens faster. If the concentration in both are the same, in both compartments are the same, so if I have 100 bouncy balls here, 100 there, then diffusion will happen randomly, but net diffusion won't. Does that make sense? Because there's no gradient, there's no driving force. The size of the molecules. So smaller molecules will move faster or have a larger net diffusion. So if you think of those bouncy balls, right, it's more likely that those bouncy balls will be able to get to the other room. But if I have those yoga balls bouncing everywhere, for it to exactly fit through that door without hitting the frame and coming back, it's going to be less probable, right, that it's going to end up in the other room. And then distance. So diffusion distance is probably the best or the strongest factor affecting net diffusion. So the larger the diffusion distance, right, the thicker you have those membranes, right, then the smaller net diffusion force, okay? So we want to have very small diffusion distance so that the movement of those, the directional movement of those molecules can happen effectively, can happen in a timely manner. And that's why capillaries have only one cell, making that capillary wall. That's why the alveoli in your lungs have only one, they're only one cell thick. Again, the membrane is made of one cell, thick only, not one cell, but like just the thickness of one cell. Small intestine as well. So all these things have very thin layers because that decreases the diffusion distance and increases diffusion rate. All right, so these are all the factors affecting that the rate of net diffusion, how fast those molecules can move from compartment to another. Water is no different. So water will move from where we have a lot of water to where we have very little water, okay? So it moves down its own concentration gradient. However, measuring the concentration of water is very hard, right? So we normally talk about the movement in water in terms of the solute. Solute are the particles that are dissolved in that liquid, that water. Water would be the solvent, okay? So we talk in terms of the concentration of the solute to indicate the direction of movement of water. And let me get my pen here really quick. So here, for example, we have a solution that has less solute. That means it's going to have more water, okay? And here I have a solution that has more solute. That means less water. So water is moving down its own concentration gradient, but we refer to it in terms of the solute. So with the solute will be the opposite, less solute to more solute. Does that make sense? Another way of thinking about it is if you think of the water needing to dissolve that solute, okay? So if we have more solute right here, then we need more water going to try to dissolve that solute. Osmolarity refers to the number of particles in solution, okay? So osmolarity is the number of particles in solution. Osmolarity is going to be osmoles per liter. You can talk about also millismoles per liter. Depending in biology, we normally talk about milliosmoles rather than osmoles. And one thing that I want to emphasize is that osmolarity describes the number of particles in solution, not the composition of those particles. Okay, so it's very different from molarity in that sense. What do I mean by this? I mean that 1 milliosmolar solution could be composed by glucose or by sodium or by chloride or a combination of all. Whereas molarity refers to a specific chemical species, right? 1 millimolar glucose, right? But you can have 1 milliosmolar solution in which you have glucose and sodium chloride in it. Does that make sense? So osmolarity refers to the number of solute particles dissolved in that solvent, so in solution. You don't care what the particles are, just the number of particles that are there, because the number of particles are what's going to create an osmotic pressure, and I'll talk about osmotic pressure in a minute. Now, you have two types of components, non-electrolytes and electrolytes. Non-electrolytes do not dissociate when you place them in water. So for example, sucrose, glucose, urea, you put them in water, they don't dissociate. They remain as glucose or they remain as sucrose or urea, right? So the molarity and the osmolarity here are going to be the same. So if you have 150 millimolar sucrose, that's going to be 150 milliosmolar sucrose because it's not dissociating, so the number of particles, of solid particles, is still 1. Electrolytes are different. Electrolytes dissociate in solution. So if you put sodium chloride in water, that's going to dissociate into sodium and chloride. So now you have 2 solute particles. Okay? Calcium chloride, that dissociates, you put it in water, it dissociates into 1 calcium and 2 chloride. So now you have 3 solute particles. So in this case, the molarity is going to be, the osmolarity is going to be the molarity times the dissociation number. So if you have 150 millimolar sodium chloride, you put it in water, dissociates into sodium and chloride, so the dissociation number is going to be 2. So the molarity times 2 is 300 milliosmolar. Does that make sense? What about calcium chloride? So if you have 100 millimolar calcium chloride, what would be the osmolarity? 100 millimolar calcium chloride. So calcium chloride dissociates into what? Into 1 calcium and 2 chlorides, right? So what is the dissociation number here? 3, perfect. So then the molarity, which is 100 millimolar times 3, which is the dissociation number, equals 300 milliosmolar. So always, if you get a problem in the final and you get given millimolar, convert it to milliosmolar. Okay, that's the first step you need to do. Convert it to milliosmolar, because that's what's going to indicate the number of solute particles and the osmolarity. Now, one number that I want you guys to remember, and I'm going to erase this really quickly, is 300 milliosmolar. So most fluids, the osmolarity of most body fluids is going to be 300 milliosmolar. So for example, the osmolarity of plasma, which is the fluid part of your blood, is 300 milliosmolar. Okay? Most cells are going to be 300 milliosmolar, the intracellular fluid. Okay? So there are exceptions, and you'll see that in the kidney, but for the most part, body fluids are going to be 300 milliosmolar. So these numbers you do need to know. All right, you can compare osmolarities of two solutions, okay? You can look at the two solutions, calculate the osmolarity or modify from millimolar to milliosmolar and compare the osmolarity of the two solutions. Isosmotic solutions, iso means the same. Osmotic refers to osmolarity. So isosmotic solutions is when you have the two solutions have equal number of solute particles per volume. Remember, it's the number of solute particles per volume. Okay? So let me give you an example. If you have a solution A that is 2 millimolar sucrose and a solution B that is 1 millimolar sodium chloride, first thing you're going to do is convert that to osmolarity, right? So sucrose does not dissociate. So 2 millimolar times 1 equals 2 milliosmolar. So that's the osmolarity. Okay? Solution B is going to be 1 millimolar sodium chloride. You put sodium chloride in water. Sodium chloride is going to dissociate into sodium and chloride. You have two solute particles. So the molarity times 2 gives you 2 milliosmolar. And then you look at the osmolarity here, and they're the same. So you say that A is isosmotic to B, or B is isosmotic to A. Okay? Both solutions have the same osmolarity, meaning that those two solutions are going to generate the same osmotic pressure. All right? Have the same number of solute particles per volume. Look, they're different chemicals, right? But they both have the same number of solute particles per volume. Now, hyperosmotic solutions it's a solution that generates a higher osmotic pressure. Hyper means more or higher, and osmotic refers to the osmolarity. So a hyperosmotic solution generates a higher osmotic pressure or has a higher osmolarity. So let's say you have a solution A of 2 millimolar sucrose and a solution B of 0.5 millimolar sodium chloride. Again, first thing you do is convert it to osmolarity. So sucrose does not dissociate. So 2 times 1 gives you 2 milliosmolar. Sodium chloride dissociates into sodium and chloride, so the dissociation number is 2. 2 times 0.5 gives you 1 milliosmolar. Okay? So now you compare these osmolarities and you see that solution A has a higher osmolarity than B. Then A is hyperosmotic to B. And in that same train of thought, then B is hyposmotic to A, because B is going to be the solution that generates the lower or less osmotic pressure. Does that make sense, everyone? Yeah? Okay. Anybody needs me to repeat this? We're all good? Okay. All right. Things are a little bit more complicated in biology, okay? So I'm going to, because we have membranes, and there are different types of membranes. There are permeable membranes that let both water and solute, or the solvent, water will be the solvent, the solutes will be things like glucose, sucrose, urea, sodium chloride, calcium, magnesium, etc. Okay? So permeable membranes allow both things to move, the solvent and the solutes. All right? So permeable membranes just let everything go through. So the pathway is open. You have semi-permeable membranes in which, I'm not putting those here, but semi-permeable membranes are the ones that allow the solvent to move, so in this case water, but the solutes cannot move. And then most biological systems have selectively permeable membranes. So these ones are the ones I'm going to discuss here. Selectively permeable membranes are going to allow the solvent to move, so water can move freely. So there's a pathway for water, but the membrane is impermeable to some solutes, very permeable to other solutes, and somewhat permeable to some other solutes. So the permeability to the solutes vary, okay? And that's what most biological membranes are going to be like. Water can freely cross, so the solvent, and it depends what solute it is. Some solute can cross freely, some other solute cannot cross at all, and some other solute has a lower permeability or a higher permeability, okay? So if we have this U-shaped compartment, right? And we split it in two. So we have compartment A on this side, compartment B on this side, and we have a selectively permeable membrane. So it's a membrane that is going to allow water to move. Water is shown as these blue circles, and then we have glucose. And glucose is shown as these big pink circles. And this selectively permeable membrane is not going to allow glucose to move, so it's impermeable to glucose, but water can cross. At this point here, we have more glucose in compartment B than A, okay? And we have then less water in B because we have more solute, less water, okay? So there's a higher osmotic pressure in compartment E than in compartment A. Why? because remember what osmolarity is the number of solute particles per volume, okay? The volume so far here is the same. So we have the same volume of water. You see the level here is the same as the level here. But we have more solute particles here relative to the water, right? It's, let me get my pen here. Right, remember osmolarity. If you remember what osmolarity is, I can assure you you can do all the exercises that I'm going to give you with the kidneys, no problem. So it's the number of solute particles relative to the volume. Volume of what? Whatever fluid it is. So in this case, water. Number of solute particles will be the solutes, okay? So in this case, the solute particles are going to be glucose, and the solvent will be water, okay? And you have more glucose in compartment B than in compartment A. And because you have the same volume, right, on both sides, you're going to have more water here and less water here, okay? So this compartment B is going to have a solution that has more solute particles relative to the volume. So if the numerator is higher relative to the denominator, the osmolarity here would be higher in B, all right? Now, let's say that I give it some time. So this is at time 0. I give it some time. What's going to happen is because that selectively permeable membrane has a pathway for water, right? So this membrane right here is a pathway, and it's open for water but not for glucose. So glucose cannot cross because we don't have a pathway. Even though we have a driving force, we have more glucose here and less glucose here. We have a concentration gradient, right? Glucose would want to move to A, but it can't because there's no pathway. Remember, we always need a driving force and a pathway. But water can. And remember, if we have less glucose here, we have more water. And if we have more glucose here, we have less water, okay? So we have, or you can think it in terms of the solute, or you can think it in terms of the amount of water, or you can think it in terms of like, we have too much glucose here in B, let's go dissolve it, move water. So water is going to move, and remember, this movement is random, but we are still going to have, because there's a concentration gradient, or in this case, an osmotic gradient. The osmolarity in compartment B is going to be higher, right? Then water is going to move from A to B, down an osmotic gradient, okay? So water moves from A to B, and water moving in this direction is going to make it so that the volume here increases, all right? So that's what we had here. So higher osmotic pressure in compartment B than compartment A. We're going to have net movement of water from where we have a greater concentration to the area of lesser concentration of water, right? So this is osmosis. This is what we call osmosis. Does that make sense, everyone? What would happen, though? What would happen if, and let me get my pen again here. Oops, sorry. Oh my God, I cannot do this. Okay. What would happen if instead I have a selectively permeable membrane that allows glucose to move through? So both water and glucose can move. So I'm starting here with time 0. Let's call this time 0, okay? And I have higher amount of glucose here, lower amount of glucose in A. But this selectively permeable membrane allows both water, is permeable to water and to glucose. So what would happen initially? First step, what do you guys think? And you need to know how permeable the things are, okay? So I was hoping somebody said that. What is the permeability? So I'm telling you this selectively permeable membrane right now is going to be permeable to both glucose and water. But the permeability, how much permeability we have to a specific solvent or solute is important, right? So the permeability for water, let's say I'm going to give it a relative number. Normally you measure it in centimeters per second. So how many centimeters are covered per unit of time, okay? So, but I'm going to give it in relative terms. So let's say I tell you that the permeability of water is 1,000. It's relative term to the permeability of glucose, which I'm going to give it as 1. So this membrane is more permeable to what? Water or glucose right now? Water, right? So the permeability of water is higher than the permeability for glucose. So the first thing that is going to happen is that water is going to start moving down an osmotic gradient. The osmolarity in B is higher. Okay, so osmolarity in B is higher, is generating a higher osmotic pressure because we have more solid particles per volume, right? So the osmolarity is higher for B. So water is going to immediately start moving. Permeability is pretty high. It's going to immediately start moving. Time 1, water moves from A to B, okay? But eventually, glucose will start moving. So time 2, then glucose start moving down its own concentration gradient. We have more glucose here in B than in A, so glucose now is going to start moving from B to A. And as glucose moves from B to A, water will then, in time 3, move back to A to compensate. And at the end, when the system is in equilibrium, we're going to have the exact same number of solid particles per volume in A and B. So the osmolarity in A and B, when we reach equilibrium, is going to be the same. Does that make sense? My point here is that you need to understand if that pathway is open for different solute particles and for the solvent. So always think of there's a driving force, there's a pathway, then it can move. How fast it can move, it's going to depend on the permeability and the driving force. How large that permeability is, how large the driving force is. If the pathway is not there, in the first example I told you glucose, that membrane was impermeable to glucose, then it doesn't matter the driving force, it just doesn't cross. Does that make sense? Okay. And that's when we talk about osmotically active and osmotically inactive solutes. So osmotically active solutes are the ones that can cross because they have a pathway. The membrane is permeable to them. Osmotically inactive solutes cannot. Okay? You don't need to worry too much about those two terms. Those you'll learn later. But just understand that having solutes that cannot cross is going to affect the osmolarity of a compartment. And I'll talk about that with an example at the end. Okay. The next term I want to talk about is tonicity. And how many of you have heard about tonicity? Raise your hand. Okay, very few. How many of you haven't heard about tonicity ever? It's okay, raise your hand. If you haven't, it's totally fine. I'm assuming you haven't, so don't be shy. Okay. So tonicity is a little bit more complicated than osmolarity. It's related to, but it's a little bit more complicated to osmolarity. So only looking at osmolarity does not determine what moves. Okay? Why? Because sometimes we have those selectively permeable membranes that will not allow some solute to move. So we have that osmotically inactive solute creating an osmotic pressure, and it cannot equilibrate, right? Because it cannot cross because there's no pathway, right? So we need to discuss tonicity because tonicity is going to determine the movement of water in or out of a compartment, or no net movement of water for that matter. So very simply, tonicity refers to the movement of water given the osmolarity of a certain compartment. You normally talk about tonicity referring to the solution that you place a cell in. And this is a very standardized experiment that people do in animal and human physiology labs. It's a red blood cell lab, so they're going to grab a red blood cell, which red blood cells have mostly hemoglobin in them, carrying oxygen. And in NAS mammals, it has lost all the organelles, no nucleus. When the red blood cell matures, it loses its nucleus, its mitochondria, everything. I mean, it's just a sack full of water and hemoglobin with oxygen. That's why they don't live for a very long time. They only live in a healthy human for 120 days. And then they get constantly replaced. But you can put any cell, basically. You put a cell in a solution, and depending on the osmolarity of that solution and what the cell is permeable to what solutes can cross that cell membrane, you're going to have water moving in to the cell, out of the cell, or no net movement of water. Water is always going to be crossing, but not directional movement. So let's talk about tonicity here. If you place a cell in an isotonic solution, and normally you talk about the solution relative to the cell, but for the purpose of this class, you can, as long as you make it clear, it's fine what you're referring to, the solution to the cell or the cell to the solution. So if you place the cell in an isotonic solution, there's going to be no net movement of water. Water is going to move into the cell and out of the cell, but directional movement, it's not going to happen. Net movement is not going to happen. Net diffusion of water or osmosis, right? Net osmosis won't happen. So the cell volume remains the same, all right? So that's a solution that is isotonic to the cell. So in this case, if I were to put it in terms of osmolarity and numbers, which is easier, I don't know, I find numbers easier to visualize, I don't know why. I told you that the osmolarity of most body fluids and cells is 300 milliosmolar. So in this case, the solution is also 300 milliosmolar. So there's going to be some movement of water, but no net movement of water, so the cell volume remains the same. Now, if we place that cell, which is 300 milliosmolar, in a solution that is hypotonic, for example, 50 milliosmolar, what do you think is going to happen? Do you think, remember what osmolarity is? Osmolarity is the number of solute particles per volume. So in this case, the cell has higher number of solute particles per volume than the solution it's in. The solution is only 50 milliosmolar, whereas the cell is 300 milliosmolar. Will water move in or out of the cell? Somebody just said it, I think, around here. And it's shown right there. What do you guys think, in or out? In, right? Why? Because we have more solute particles per volume in that cell, so water would want to go in and dilute it, right? Or you can just think about this one having high water, it's not writing, high water, and this one having low water relative to the solution. So water, net movement of water is going to be into the cell. If the solution is too hypotonic, like when you place a cell in distilled water has no solutes, it's distilled, right? So it's too hypotonic, then all of that water will come into the cell and cell volume increases until it bursts. It not always bursts. It needs to be, the solution has to be very hypotonic relative to the cell. Because in that case, a ton of water will move into the cell. And I mean, you can, it's like a balloon, right? You can stretch it so much, but at some point, boom, it's going to burst, okay? Now, hypertonic solutions. So let's say I have my cell with 300 milliosmolar, and I place it in a solution of 1,000 milliosmolar. So the solution is now hypertonic. It has a higher tonicity relative to the cell. So water is going to move out of the cell because we have now more solute particles in the solution than in the cell. Okay? So water is going to move out of the cell, as you see right here. And if that solution is too hypertonic, like 1,000 milliosmolar is salt water, that's pretty hypertonic, then it's going to, all of the water of that cell is going to come out, and we end up with crenation. Crenation is when all of the water leaves the cell and the cell ends up with just the things that couldn't move, couldn't cross that cell membrane. If it's a red blood cell, it will be hemoglobin. Okay, cell, in both cases, this one and this one, the cell dies. If it bursts, it dies. If it crenates, it dies. Does that make sense? Okay, so when we study the kidneys, we're going to look at how kidneys maintain fluid balance and regulate osmolarity. So we're going to be talking about osmolarity and tonicity. And I'm going to give you an example, and they're very, very similar sometimes, but I'll try to make it clear when osmolarity and tonicity is different. So the access code is 148, and I'm going to give you an example here. So let's say I have two compartments, okay? Compartment A and compartment B. And this membrane, I'm going to actually make a membrane here. This is a selectively permeable membrane, and that membrane is going to be permeable, let's say, to water. I don't know, it's not letting me write. Let me try to do it here. Oh my God, it's not letting me. Urea and sodium chloride. Okay, so the permeability to water, again, I'm going to put it in relative terms, so 1,000. To urea, it's going to be 100, and to, this is supposed to be in centimeters per second, but I'm giving you a relative number here. And let's say sodium chloride is 0, okay? So I'm going to put a bunch of water in compartment A and compartment B, and I'm going to put here 100 milliosmolar urea and 100, actually I'm going to do it 100 millimolar urea and 100 millimolar sodium chloride. And at time 0, the volume of water in both compartments, I'm going to put it like this, is the same. Okay? So first thing that I want to ask you is compartment A isosmotic, hyposmotic, or hyperosmotic to compartment B. And I want to tell you that is before water crosses, before any water crosses. Why am I giving you that assumption, before any water crosses? Because look at the permeabilities, at the permeability table right here. Water is going to be the one that has the highest permeability. That selectively permeable membrane has the highest permeability for water. So that's, regardless of the driving force, if we have a driving force, that's what's going to move first, the fastest, water. Because the relative permeability is 1,000 compared to urea, which is 100, and sodium chloride, not permeable too. Okay? So here first, before any water crosses, what do you need to do? What's the first step that I told you need to do? Convert to osmolarity, correct. Convert to osmolarity. So 100 millimolar urea, what would be the osmolarity there? It'll still be 100 because urea does not dissociate. So 100 milliosmolar. Units are important, okay? What about sodium chloride? 100 millimolar sodium chloride is what? Do you need the... This is important, guys. You'll probably get an example like this in the final. So it's important to understand it. So 100 millimolar sodium chloride is what? 200 milliosmolar, right? Because the dissociation number is going to be 2, right? So we multiply the osmolarity, the molarity times the dissociation number. It's going to dissociate into sodium and chloride, right? So we have now 200 milliosmolar sodium chloride. Okay? So the osmolarity here is 100, the osmolarity here is 200. Before any water crosses at time 0, A is iso, hypo, or hyperosmotic to B. What do you guys think? Which one has the highest osmolarity? B has the highest molarity, right? It's 200. This one is 100. So A is iso, hypo, or hyperosmotic? What's that? Hyposmotic. So this is the answer. Hyposmotic. Okay? Now, let me ask you the second question. So the second question is A going to be isotonic? Let me change the ink color. So isotonic, hypotonic, or hypertonic when water starts crossing. So now we're assuming water is going to start crossing, so other things are going to cross as well. So we're in time 1. Water starts crossing. So water is going to start crossing where? From A to B or B to A? B to A. So water is going to move from A to B, right? Because this one has the highest osmolarity, right? So water is going to start crossing from A to B. Where we have more solutes, we have less water, right? So water is going to start moving from A to B at time 1, right? The permeability is high. What about urea? Is urea going to start moving? Do we have a driving force? Yes, we have a driving force. Do we have a pathway for urea? Yes, we do, look. So urea is going to move down its own concentration gradient. Urea is going to move from A to B, okay? But it's lower than water. So once we have urea here, urea is going to move until the concentration gradient is eliminated for urea, right? Urea doesn't care about sodium and chloride and water. It's just moving down its own concentration gradient. So we're going to move urea here, and so now we have urea. And just to make it, visually clearer, we're going to move half of the urea here. And so now we have 50 milliosmolar in A and 50 milliosmolar urea in B, okay? Can sodium chloride move? we do have a driving force, right? But we don't have a pathway. Look, permeability is 0. So sodium chloride is one of those osmotic inactive solutes. So at this point, when water starts moving and urea starts moving, we end up with an osmolarity in B of 250. Remember, osmolarity doesn't care what that solution is made of. It just cares about the number of solute particles per volume, right? And here, the osmolarity is 50. So as water starts to cross, is A isotonic, hypotonic, or hypertonic to B? And think of this over here. Think of this diagram right here. Is water moving into B, out of B? So water will move, will keep moving right in this direction. Water is going to keep moving in this direction. So water is moving to B. What do you guys think? What's that? So water is moving from A to B, right? So you have this situation. Let's say this is A and this is B. So then A would be hypotonic to B. Just look at the osmolarity. This one is 250 and this one is 50. But now we're only talking about the movement of water. That's why we talk about tonicity. So water keeps moving from A to B. A is going to be hypotonic to B. So the same as this scenario here. Does that make sense? So the things that I want you guys to do, it's almost time to go. First things first, you convert everything to osmolarity, step 1. We can do another of these exercises. If it's confusing you, we can do it on Wednesday. First things first, you convert to osmolarity, okay? That's the first thing. Once you have the osmolarity, you're going to look at the permeability, okay? And determine what can move and what cannot, okay? First question you're going to get is something iso, hypo, or hyperosmotic to another compartment. So is A iso, hypo, or hyperosmotic to B before any water crosses. So at that point, nothing has moved. You just look at the numbers, right? So we had here 100 milliosmolar urea, and here we had 200 milliosmolar sodium chloride. Before anything moves, which one has the higher osmolarity? B. So we know that A is hyposmotic to B. A has a lower osmolarity than B, okay? Now, second question you get is when water starts moving, and therefore other things are going to start moving, is A iso, hypo, or hypertonic to B? That's when water can move is when I'm going to talk about tonicity, not before, okay? Because tonicity really refers to the movement of water relative to the osmolarity. And that's why it's a complicated topic. So if you're not understanding tonicity right now, that's totally fine. We'll get it, okay? It'll make sense on Wednesday. So what you need to do now is have water move. So water starts moving from A to B because B has a higher osmolarity. Nothing else has moved. That's time 1. Time 2, whatever solute the membrane is permeable to, it's going to start moving down its own concentration gradient. In this case, the membrane is permeable to urea. You have 100 milliosmolar urea in A, no urea in B. So urea is going to start moving down its own concentration gradient until it equilibrates. So you're going to end up with the same amount of urea in A and in B, right? Sodium chloride cannot move because the membrane is not permeable to it. Even though we have a driving force, not permeable, so it cannot move. So you end up in B with urea and what you had for sodium chloride. You add the two osmolarities, 50 milliosmolar urea plus 200 milliosmolar sodium chloride, that gives you 250 milliosmolar. And on this side, you only have 50 milliosmolar. So the osmolarity there is higher in B. Water is going to move to the compartment with the higher osmolarity. So water keeps moving from A to B, right? That means that B is hypertonic, or A, in the case of this question, A is hypotonic to B. Does that make sense? A little bit more? How about on Monday? On Wednesday, I'll give you another example before we start the kidneys. Good? But you guys solve it. I'm going to have you solve it with a partner and try to really solve it because one of these guys is going to be in the final.